Perfect.
Yeah, thank you.
Thank you so much for this opportunity to speak here at this drug development meeting. And I would like to talk about something that is probably not in the direction of the majority of us that have been working in drug discovery for many years. And this is everything related to molecular biology and the obviously seismic change that has been taking place in this space. In the early 2000s, we were able to sequence 1,000 base pairs per day. Today, we're able to sequence that amount of base pairs in 1 second. The FDA has approved about just a handful of gene therapies right now, but there are over 400 clinical trials in gene therapy with over 500 companies working in this space. So we're talking about a real revolution on an area that I think is absolutely relevant for the future of medicine. I am the Chief Medical Officer of ASC Therapeutics, a company that is focused on gene therapies, Crispr therapies, and also cell therapies. And I'm very honored to have a chance to talk to you about my work and also my perspective in this space. This is the outline of my presentation, where I'm going to start describing current drivers on gene therapy drug discovery, the relevance of biomarkers in gene therapy drug development in 2 areas. First, on selecting the patients and then in monitoring the patients. And obviously that is absolutely relevant because you need those tests during the process of drug discovery. And I will give you a reference or a benchmark on the work that we are doing at our organization on Hemophilia A with gene replacement of factor 8. And when after having done all that, I would like to spend a moment about the future drivers of this space. Let me start with current drivers of biomarkers in gene therapy. It's not only the fact that we are right now able to search and identify genes, we are also able to identify their function, and we're able to genetically engineer them. So we have rediscovered genes responsible for over 5,000 rare genetic diseases, but there are over 100,000 genes associated with common diseases from diabetes to heart disease. So this is the direction that an area of medicine is taken. And the way we can assure that all these advances are useful for patients is ensuring that precision medicine is applied to that and changes the way we think about prevention, treatment, and monitoring of many of these diseases. For that, we need biomarkers. So we need to understand how these therapies, very precise already, are really applied to the right patient at the right time. So we need to identify what patients are going to be the ones that should be selected for this therapy, and then monitoring these patients once they receive that therapy. Now, this monitoring is relevant for the patient, but it's also very relevant for the actual progress of this space. When talking about several biomarkers in this area, there are several types of biomarkers of disease. They could be related to the immune response, cellular response, or molecular response to these gene therapies. And we need to identify, and you're going to see some details in a moment, the way these therapies do mainly 2 things. Number 1, the way they incorporate into the cell, and second, the way this cell starts becoming a biofactory of the therapy, which is the gene that we are trying to replace. So therefore, it's extremely important to select the right patient to the right therapy and monitoring this patient's response as a way to really advance drug discovery. So let's now spend a moment on those biomarkers. And I'm going to take a stab at just the concept used by the FDA on how biomarkers, which is a statement from the FDA, the biomarkers provide information essential for the safe and effective use of a corresponding therapeutic product. So what are those biomarkers? There has been a lot of work done, mostly in the academic space, clinical settings, industry settings, on what is called omics, because there are a number of notions that are genomics, understanding the genes, transcriptomics, how these genes are have this transcription into our RNA, epigenomics, proteomics, phosphoproteomics, metabolomics. So there are a number of ways to assess and to identify those biomarkers. And these biomarkers obviously can be single biomarkers. They can be a network of biomarkers that work together. But what is more relevant about what is going on today is the potential to develop signatures. And these signatures are driven by machine learning and artificial intelligence methodologies that are able to identify what combination of biomarkers are adequate for what patients. So you identify your patients, you identify your type of gene therapies based on biomarkers, and then you apply that to the different patients. This is targeted medicine. So the biomarkers can be used in two ways, as I mentioned just a moment ago. First, they can be used to select patients. And the most obvious way to say that is that if you have a monogenic disease where there is a gene that is faulty, you replace that gene. And that's the most obvious way to initiate a gene or even a cell therapy. So what you do is that you genotype the target genes or phenotype the target cells. And once you do that, then you start looking into the functional effects that your new replacement gene is going to have on target systems or organs that replicate the physiological environment in an individual. Once that has been the case, then the next step is to monitor the patient response over time to ensure that we achieve the maximum patient benefit. So that will help us to titrate the drug and to ensure that we also define what's the most targeted dose for that individual. So at the end of the day, while gene therapy is already very precise in the sense that you are targeting a well-known gene and you understand well also the function of this gene, and we have this type of advanced biologic therapies with an intrinsic benefit of being precise tools for manipulating human biology. They're just reserved to those cases where we indeed understand the pathophysiology of that disease. So once we get to that step, the next step is to monitor the patient response over time. And for that, we have two types of biomarkers. First, efficacy biomarkers that are related to the gene or cell product clinical activity. And secondly, safety biomarkers that are going to indicate us the expected adverse events. Obviously, a biomarker does not replace efficacy endpoints that are built into the actual clinical trial. And that's my job. As chief medical officer in my organization, I design studies, I conduct the studies, and at the end of the day, what the agency, what the FDA or other government agencies are looking at is how are we going to be able to identify that this drug is working. And biomarkers are a great surrogate indicator of that activity, also in terms of safety, but even more important is to be able to hit the endpoints that are predefined in these clinical trials. So let's now, instead of me giving you more theoretical kind of indications about how everything works, I'm going to give you an example about the gene therapy that we are doing at the ASC Therapeutics. And this is a case study about Hemophilia A. First, let me just give you a perspective about what is going on in this space, about molecular medicine and how while we start discovering or we started discovering, and still do obviously, with the small molecules, we can potentially go into large molecules. I worked in the large biologic space, being part of the team that developed the Humira drug for a number of indications. There are also even additional complex molecules with the so-called pegylated protein. And you can see at the bottom kind of an example of how to find a kind of reference. And if all these extraordinary molecules that were used, monoclonal antibodies mostly, or complex with pegylated proteins, were kind of jet fighters and Air Force One, we're now moving into an international space station. Gene therapy are very large molecules. They are viruses that contain the genetic material that is going to lead into gene replacement. And they contain so many elements that we can consider them as a so-called international space station if we see the progress in terms of technology. So this has a tremendous impact, especially in moving from drug discovery in the lab to pre-IND enabling or IND enabling studies, to the moment that we start scaling up and producing drug for clinical trials and then for commercial settings. The complexity is tremendous, and the number of variables that are incorporated here are out of this world, I would say. And for me, being a chief medical officer focused on clinical trials, I would say that I was not expecting to really be involved so much in manufacturing, in CMC, and understanding also all the process that leads into the moment that they get a vial that can be used and can be infused into a patient. So Hemophilia A, this is the reference I'm going to be speaking about. This is a disease where you have, in this case, is factor VIII, which is faulty. These patients are not able to complete the entire coagulation cascade, and they have a lot of bleeds. And some of them are very serious because they can happen even in the joints and lead to a situation where this patient ends in a wheelchair. But there are many other serious situations. And by the way, what happens in these patients is that in some cases, because they need the replacement of factor VIII with synthetic or with animal derived factor VIII, they develop inhibitory antibodies. So that's what you see here. And it's a large percentage of patients that develop them. So it's very important to understand that that's already a biomarker itself, because we want to see which patients have already an antibody that is going to inhibit factor VIII, no matter how you're producing it. So if you produce this factor VIII in the cells of the individual, of the patient, still it's going to be inhibited. So that's why it's very important to understand that and use that as a way to select patients. This is a large unmet need. These patients require visiting the hospital every several times a week sometimes. And when we develop, and we are not only our company, but others are working on gene therapy, we're talking about being able to have one single treatment of these patients, a couple of hours, injecting them these international space stations, and they will not require to visit or to receive therapy anymore. It's one and done. So this is what gene therapy, why gene therapy is so important, and it's already having some patients. And I work with the Avexis team, with the SMA, spinal muscular atrophy, with the SMN1 gene, and we were able to replace that. That company was acquired by Novartis by almost $9 billion, and it has changed the lives of many patients that otherwise would simply die of this terrible disease. So the objective in our case is to inject a viral capsid with a transgene and with type of combination of a virus, which is a harmless virus that has tropism for the liver, and in the liver cell, it incorporates into the cell, into the endosome, and then it releases the transgene into the nucleus. And this is the circular kind of color that you see here, which includes the transgene, which is factor VIII, and also a promoter, which is a sequence or a DNA sequence that is going to promote the production of the gene, in this case factor VIII. And as you can see, separated from the chromosomes, you see the green chromosomes there, the liver cell DNA. This construct is not integrating into the actual DNA of the individual. So it's in some way safe. And this is relatively similar to what happens with the mRNA COVID-19 vaccines. In that case, we're not giving a DNA like is happening here that goes into the nucleus. The mRNA stays in the outside of the nucleus and starts producing the protein, which in this case are the spike proteins that are going to defend the individual from COVID. So I like to use as a kind of reference or analogy the historic flight to the moon, where Apollo 11, excuse me, went from the Earth to the moon, and they had a number of components. Number one component was the actual rocket, and the rocket at the top contained the lunar module and also the capsule, which also had the potential for fueling also the entire trip, let's say, from the Earth to the moon. And the rocket would be, in our case, the virus. The capsule would be the promoter that I mentioned a moment ago, and the lunar module would be the actual transgene that we're going to be using. And here you have the way it works in our case, with, again, the entire machinery of the AAV, which is the type of virus that we use. And in our case, what we're using is, and you can see to the right, upper right, we have the capsule, which is this promoter. It's an engineered liver-specific promoter, minimizes the size of the payload that we're going to incorporate into the rocket or the LAV virus. And we also have the liver-engineered transgene, which you see here a depiction of it. It's missing one domain, which is the B domain, but we have it in a format that is extraordinarily efficient. And in our case, what we have here is transgene, which incorporates 9% of sequences ultra-selected from the porcine factor VIII, which increase the transcription, translation, and secretion of the factor VIII. So we're improving the efficiency of the liver cell to manufacture our drug, which in this case is is factor VIII. So all this has been very nicely engineered, so everything works together. So what we have been seeing in preclinical data, and we're about to initiate clinical studies, phase 1 clinical studies, is that we could clearly see that with this construct that I mentioned a moment ago, we are able to significantly elevate the factor VIII protein levels in a dose-dependent manner. That's what you expect. And with that, now we are looking into analyzing the potency of our construct. First, we need to see how it what was the capacity to infect liver cells. In some way, you are producing a kind of very mild hepatitis, absolutely harmless. But then you have also to measure what I mentioned a moment ago, the potential protein inhibitors that may impact the factor VIII protein levels. And very important also is that at least in animal models, we were not able to observe breaking adverse events. So even using non-human primates. So we are going to measure all these in our clinical trials, and we do not expect to have any indication of failure. And that's also because a number of other companies are already working on similar constructs. So besides what I mentioned before, there is also another approach that we're taking. It's a different program, but is related. And this is using CRISPR. So what we do with CRISPR, and you see here scissors, is that we cut at a very selected point on the genome of this individual, and we insert our construct. In this case, is integrated into the chromosome. It's not what I showed before in this case, and that's why you see at the upper right, Apollo 11 landing in the moon. We are landing in the chromosome. We are really incorporating this construct in a way that the body itself is going to use its own promoters. We use the albumin locus in our case. We don't need a promoter. But the advantage is that this correction of the factor VIII protein, this transgene, is stably integrated in the genome. And this is why it's an advantage for pediatric patients. Because children have a lot of turnover and cell division in the liver, so that if we use the previous approach, which works very nicely in adults over 18 years old, we would not be able to use that approach. So in the case of children, we can integrate factor VIII in a way that is stably manufactured forever. It takes, again, we use the albumin promoter, and it's a very site-specific integration of the genome. So what you don't want to see, and this is also a problem of CRISPR therapies, could be that you integrate these genes in off-target areas, and this is not the case because we use a very specific and a lot of screening to find the right way and the right approach. So in general, in hemophilia A, as a summary, remember what I mentioned before, there are selection biomarkers, there are monitoring biomarkers. We need to make sure that we generate a factor VIII activity that definitely goes above 2 units per deciliter, so that these patients are going to be able to avoid all types of bleeding that can come from different sources. The selection biomarkers in this case, which we have incorporated into our program, are inhibitory antibodies of factor VIII protein, neutralizing antibodies to the actual virus that we use as the rocket that transports the transgene to the cells. We need to understand how the liver is functioning, and we have imaging and liver enzymes that tell us about the health or the damage of the liver. And we also have factor VIII gene mutations, which are different in each individual. Depending on that, some patients are able to produce already some minimal amounts of factor VIII versus others. Now, once these patients have been selected, they start in the clinical trial, then we monitor these biomarkers. And the most important aspect from the agency perspective, and from my perspective as a clinical developer, is to ensure the safety of the patient. So I need to ensure that there is on-target liver AAV activity, this virus that we use. We have to exclude off-target of other organs and tissues. We look into the total neutralizing antibodies to AAV8 once the patient has received that virus, and we look into the cellular immune response as well as the factor VIII inhibitor levels. In terms of the efficacy, is well established the way you do that. You look into factor VIII activity. We have a number of tests that are able to tell us how this patient is able not only to produce the protein, but what is the activity, the functionality of the protein that we have given to this patient. So in terms of future drivers of biomarkers in gene therapy, it's very clear that we really need to continue understanding and working on the biological underpinnings and genetic drivers of disease. It's like a vicious circle, in this case, where you have a positive feedback from the data that you obtain with patients in order to develop a better and more efficient drugs. And that's why through these kind of therapies, which are so targeted, we are able to understand better and better the way human biology works around this disease and around this defect. Now, we have nowadays, and we are seeing even more and more advances in next generation sequencing and computational biology that is going to allow us the discovery of disease mechanisms on those diseases that we're already treating and future diseases. And finally, everything related to big data. Everyone knows nowadays that the amount of information that we're collecting from each and every patient, the amount of information that comes from natural history, from the way patients are treated with other therapies, even, we're going to be able to generate predictive models of gene expression and regulation that will help us understand the complex mechanism of disease. I mentioned just a moment ago how it's relatively easy to treat patients with monogenic diseases where we understand clearly where was the defective gene. But if we start moving, and we will, into diseases that have a very complex genetic underpinning, which diabetes, heart disease, we will need this kind of predictive models to understand what is the role of the different genes and how they interact together to lead into those diseases. So with that, I thank you everyone for your attention. and I'm happy to take any questions.
Yes, thank you so much, Dr. Oscar. Anyone has any questions from audience? Yes, you can unmute Mr. Don Turner, you can unmute yourself.
Hey, Don Turner here again. Fantastic job. You did a wonderful job at really painting the future and the potential. I love the reference to the International Space Station, just, compared obviously to the bike. Sometimes you get lost in the lingo and you lose the frame of reference, which is really, really important. So, great job. Again, wonderful job at presenting what the future could look like. And kind of with that being said, what are your thoughts on the potential to utilize gene therapy for neurological disorders, neurodegeneration? So, part one of the question is, what's the future there that you see? And then two, more importantly, what do you see as some of the challenges in dealing with the blood-brain barrier in delivering gene therapies for, again, neurodegeneration?
Excellent question. Absolutely relevant and exciting to me because I am a physician scientist, and when I started working and when I completed my PhD work, my focus was on the blood-brain barrier in neuroinflammatory diseases. Wow. I was working on chronic polyradicular neuropathy. I know that's even still difficult for me to pronounce. And another is Guillain-Barré syndrome, which is a peripheral type of inflammation that has been also observed in some patients with COVID. But independent of that, and I could spend hours talking about the blood-brain barrier. The first thing that we need to take into account is that these viruses that we're using, this AAV, are able to cross the barrier and are able to look for the actual target, which, let me give you an example. A company I work with, which is Avexis, where you have a disease that is spinal muscular atrophy because of the lack of a gene called SMN1. And this gene is defective in the motor neurons. So there are the neurons that really drive respiration, they drive movements, and because these patients lack this protein, they die before 1 year, or being 1 year old, and they end in ventilators, and they are not able to survive. All this changed when Avexis developed, together with the Nationwide Children's Hospital in Ohio, the treatment with Zolgensma, which again is now commercialized by Novartis worldwide. And I was able to see how patients really in videos, because that's what we presented to the FDA. When you conduct these studies, you are not, you don't have a placebo control arm. You just treat patients and then you use natural history. So all patients that receive, the majority of the patients that receive this gene therapy, were able to start walking around. And there are now pictures, there are videos, or there are follow-up of patients. And the time I was working with Avexis was 2015 and 16. So now since 5 years, some of these patients are 7 years old, and they have a normal life. So that's my answer to your question. It's real. We are able to go into this central nervous system. We are able to incorporate these genes into motor neurons, and we are able to produce this protein that is making these patients absolutely normal and cure them. So that's the way we see that. We don't see any decline, at least in the past 7 years, in these patients, but obviously you need more and more time. And that's also what happens with Hemophilia A. There are some indications that there might be a problem with durability of the therapy. So that's an issue. But the direct answer to your question is yes, it works. And there are a number of patients or companies working on other neurodegenerative degenerative diseases, all in the central nervous system. And they're using a different AAV, a different type of virus. They're a family of viruses. We use AAV8 for the central nervous system. It's better to use AAV9. So again, these are details of what we do in our business.
And thank you so much. In that particular drug for the neurodegeneration you referred to, how is that administered?
An infusion.
Infusion.
So it usually is systemic venous infusion, but there were some work also looking into better delivery approaches directly into the spinal fluid. So there were some studies looking into that, but it's not necessary. So you can still do that through a regular systemic infusion. A couple of hours and that's it. The patient is.
And then my last question, very particular around Alzheimer's. Your thoughts on potential use for Alzheimer's?
I think that Alzheimer's is probably a very complex disease. It's at the level of diabetes or heart disease. So this is not a monogenic disease, so it's going to be very difficult. And the problem is that that's a disease that has been going on over time. And you saw what has happened with Aduhelm, which is this drug.
Yeah, Biogen disaster.
And the disaster, I mean, at the end of the day, no one knows why they approved it, why it's out there. And yes, it reduces the plaques, but it seems that it's not clear that it has a long-term effect on patients. I don't think we are going to be able to have a gene therapy for this type of disease or similar diseases, unless you have, and there are some, like ALS, that probably are more dependent on one or two genes, are more